Okay, today we are going to be looking at how DNA is used as a genetic code and how that code kind of leaves the nucleus so it can find a ribosome to make proteins. So DNA, remember, is just an information molecule. It has instructions to make everything inside the cell to control what happens inside the cell. So DNA, remember, is our blueprint, and it's actually proteins that carry out the work, are Bob the Builders, right? So DNA is not used directly to make proteins. It needs to be translated. And how it's translated is through a molecule called RNA, ribulose nucleic acid. Okay? So RNA is needed to make proteins. It serves as this intermediate molecule because DNA never leaves the nucleus. And where do you find ribosomes inside of a cell? Well, if you remember, ribosomes are in the cytoplasm, or they're on the endoplasmic reticulum. So they're not inside the nucleus. So how can DNA carry these instructions, these recipes for making everything inside the cell if it never leaves the nucleus? Well, the answer is a molecule called messenger RNA, or mRNA. So this is messenger RNA, okay? And you can kind of see that it looks similar to DNA, but it does have some differences, and we'll talk about those differences coming up here. But there are some other types of RNA that are involved in making proteins. Ribosomal RNA, or rRNA. And so this is my ribosome. It's made up of rRNA. So if I ask you a question, what a ribosome is made up of, you would say ribosomal RNA or rRNA. Another molecule that's involved in the production of proteins is called transfer RNA or tRNA. And it kind of looks like this broken dynamite structure. Or maybe you think of a granny hunched over with 3 legs. I don't know. But the purpose of these RNA molecules is they're going to work together to make proteins. Okay? RNA carries that information from DNA to the ribosomes. It's the messenger. And remember, ribosomes are the organelles that make those proteins. So mRNA is going to be made in the nucleus. It carries the message or the recipe or the instructions for a protein to be made. It's going to find a ribosome outside of the nucleus. And then these transfer RNAs, what they're going to do is they're going to carry amino acids to make that protein. So here's the flow map of how DNA is translated. DNA, which is in the nucleus, pretty much stays there. But when we make RNA, this intermediate or go-between molecule to proteins, that process is called transcription. And so today's lesson is going to be about transcription, and the next lecture will be about translation, taking that RNA and actually making that protein. So today we're going to focus on how does DNA make RNA, or how is RNA made from DNA? It's actually quite simple. Okay, the process of transcription just means we're going to go from DNA to RNA. What happens is that our double helix DNA molecule, our spiral staircase, is unzipped. And one side is going to be used as a template. But the base pairs change a little bit. Remember, with DNA, we have 4 bases: adenine, thymine, cytosine, and guanine. A pairs with T, C pairs with G. Well, in RNA, we see some of the same nucleotides or bases, but one changes. So we have C pairs with G, but now we have adenine pairing with a molecule or a nucleotide called uracil. And I guess maybe I should type out uracil. Okay, A pairs with U, uracil. So our T's become U in RNA. And that's one way that you can tell the difference between DNA and RNA, is that the bases between these one is different. Here we have all tigers can growl, but in RNA, now we have all unicorns can giggles. A pairs with U, C pairs with G. Now, DNA is unzipped. One side is going to be used as a template. Okay, we call that template that's being used, that's the side that's being transcribed. So only one RNA is going to be produced. After that RNA is produced, the DNA molecule closes or zips back up. So DNA unzips, one side is used, nucleotides come in, they match complementary, and then once it's done pairing up, then RNA kind of moves off to the side and DNA zips right back up. So if that's confusing, don't worry. I guess we'll model it here slowly. Okay, let's first off see if we can practice matching complementary to DNA. So the bases are going to be the same. Okay, so what pairs up with C? You'd say G. I'm just going to slide this over. And what pairs up with C? G. What pairs up with A? Thymine. What pairs up with T? Adenine. Pairs up with C? G. Pairs up with G? Cytosine. Another G means cytosine. A pairs with T. T pairs with A pairs with T. C pairs with G. And T pairs with A. Okay, so I was just practicing matching complementary. This C stands for complementary DNA. So here's my DNA, okay? Now let's say that our DNA is unzipped, all right? And we're going to use this strand to make our rRNA. RNA. Sorry, I don't know why I said rRNA. RNA. So it's going to match complementary, okay? But A is going to pair with U's, and T's will pair with A's. All right, so here we go. Starting with C. C pairs with G. C pairs with G again. Okay, so T pairs with A, so nothing, that's normal. But all of a sudden we have an A. And now remember, we're making RNA, so there are no T's in RNA. So T's become U's. So we're going to have a U there. And then G pairs with C, and C pairs with G, and C pairs with G again. T pairs with A pairs with U, right? T pairs with A, G pairs with C, and then A pairs with U. There are no T's in RNA, so we just made our RNA molecule. Okay, here's another example. Let's, again, match complementary. And I don't know why this got shoved down. Oh, shoots. Well, anyways, let's match the complementary DNA underneath it. This is supposed to be like lined up with this, and I don't know why it's not. So, G pairs with C. A pairs with T. We're still in DNA here. G pairs with C. This is a T. A, T, C, G, G, A, T, T. All right, now let's do our RNA. So C pairs with G. I'm just going to move this. T pairs with A. G pairs with C. T pairs with A. All right, A pairs with U. T pairs with A. C pairs with G. G pairs with C. G pairs with C. A pairs with U. T pairs with A. T pairs with A. So just made our RNA molecule. Okay, so this is how it goes down, right? The first step is that DNA is unzipped. So I'm going to unzip my DNA. Okay, and let's just make the mRNA molecule from the top here, okay? So one side's going to be used as the template, and we have these nucleotides that are just floating around in the nucleus that are going to match complementary. So G pairs with C, T pairs with A, C pairs with G, A pairs with U. We don't have any T's here. G pairs with C, C pairs with G, T pairs with A, G pairs with C, A pairs with U, and then T pairs with A. So we've made our mRNA molecule here, okay, and it kind of leaves the nucleus. So I'm just going to get it out of here. I'm just going to move it down here, and then DNA zips right back up. Okay. Let's say that we wanted to make the mRNA molecule from the bottom strand. So first step, DNA is unzipped, and now we have these nucleotides that are going to match complementary to it. So what pairs with C? G. What pairs with A? U. What pairs with G? C. What pairs with T? A. What pairs with C? That'd be guanine. What pairs with G? Cytosine. What pairs with A? uracil pairs with C. Guanine pairs with T. It'd be adenine. And the last one pairs with A. Uracil, U. Okay, so I've made my mRNA molecule. It's going to leave the nucleus to find a ribosome, and then my DNA zips right back up. So those are basically the 3 steps of transcription. DNA unzips, nucleotides come in and match complementary. I don't have T's, I have U's now. Once that mRNA is made, it leaves, and the DNA zips right back up. So you do have homework questions, C questions, the 3 kinds of RNA bases. Okay, there are 4 of them, but remember one of them's different in DNA, versus DNA. Describe transcription, basically the 3 steps. Give me the RNA for the following DNA code, and then define transcription. Okay, so that does it.